Hi everyone, so we're going to pick up where we left off in part one, where we kind of touched on Mendelian genetics and how that works. Today we're more going to talk about genetic inheritance, that process, specifically meiosis, and how genomics kind of factors into that. Keeping in mind with this class, DNA is the focus, and it's kind of this blueprint, but this is kind of going to start having that conversation about why DNA is not the end-all be-all. So if you want to look at ancestry, your mitochondrial DNA, not your, strictly human DNA, is actually one of the best places to start because it only comes from your mother. This is because during meiosis, the female egg is large and it has enough space for mitochondria. Mitochondria have their circular DNA genomes. They're not technically plasmids, they're just circular bacterial proto-DNA. Remember, this class isn't touching on mitochondria being endosymbiotes. That's a whole nother fun conversation. But they do come complete with their own genome, and it's about 16,000 base pairs long, I believe. So the cool thing is that only your mothers pass this on, so we can track it very easily. Okay, it doesn't get dispersed, it doesn't get mixed. It's a single line from your mom, and her mom, right? So as Homo sapiens emerged from East Africa, as we can see coming through here, I don't want to draw in here and obscure some of these haplogroups, we start to see genetic divergences. And the best way to track this in a lot of cases is mitochondrial DNA. And we'll see that's because everybody has a mom. We'll see that there are other pretty strict ways to track ancestry, but they're not quite as clean. So in these populations, you see that as humans spread, we can track strictly mitochondrial DNA in a lot of cases to see how that happened. And one of the cool things is that everybody alive today can track their mitochondrial DNA. At least we all have at least the segments in common that we all have, all track back to one Homo sapiens woman. Very far back, I think something like 200,000 years. Oh wait, that's a little much. I think 20,000 years ago. You'll have to check me on that. Lots of Homo sapien women were alive at that time. But as you can see, if those ancestors, like we have in purple here, if they didn't have any offspring or they only had male offspring that cannot pass along the mitochondrial DNA, then that line ended. But we can see, based on the Neanderthal and Denisovan genome, that Homo sapiens mitochondrial genome all have common sections that go to this, what we call mitochondrial Eve. This single woman that, to this day, is kind of the, not last common ancestor of humans, but it's the final trace genetically that we can map to. So it's very interesting. Like I said, lines end when there's no, when you have sons. So in my case, I can't pass on the mitochondrial DNA from my mother, and I don't have any male siblings, so that's it, You can track in a similar fashion the Y chromosome DNA. So remember the Y is for, is the male gene. And this is great and fine, and it's a very good way to see paternal haplogroups, but this can only be used to track the ancestry of half the population. You can only look at you if you're a male, number one, and then your father, his father, and so on and so forth. So it doesn't do the same job. It's not as good as mitochondrial DNA by any means. So the other more raw way of tracking ancestry are things called single nucleotide polymorphisms and indels. So what a SNP essentially is that in two distinct individuals, so we have 1 and 2 right here, some people have different combinations of DNA at the same region of the genome. So if you noticed all the other red right here, these are all the same between these two individuals, and these are conserved regions in the human genome. Most of us, listening to this, other humans, we have a massive amount of DNA that is pretty much the same between us. The two main ways that we have raw differences are SNPs. Sometimes these can be in genes. And like we talked about, that's like alleles. Alleles are essentially SNPs that actually cause damage to a gene. SNPs are even better for tracking ancestry, though, because these are in sections of the DNA that are not coding into a protein. So we can't use alleles as a good track of ancestry because alleles change genes, and thus they change evolutionary trajectory, right? They could give an advantage or a disadvantage to a population. SNPs don't give any advantage or disadvantage because, well, as far as we know, now we'll see why that's different. That's because they don't change an actual gene. They're just basically leftovers, right? They're just mistakes that your DNA makes while it's replicating along the way, and we can track that. An indel is similar in that it's not a changed sequence like we have here. Just between two people, somebody's just lost it. They've just lost those two DNA regions, or these two DNA nucleotides right here. And remember, since this isn't in a frame in DNA that's not coding, it doesn't matter that you've lost one space here. It's not like a frame shift mutation that you would see in a coding. So the way that we started to revolutionize being able to track DNA was polymerase chain reaction. I will put a link to this video because it's a really fun animation. PCR, as it's called, is a way for us to selectively amplify specific regions of DNA. So if we wanted to sequence these two sections of these two people, we would perform PCR within this region right here for individual 1 and individual 2, and we would be able to see, okay, there's an A and a T here, there's a G and a C in this region, that means these two are genetically different, right? I am overly ambitious, but I am going to try and do polymerase chain reaction like I was on a board right here maybe. Do forgive me if this doesn't go well. Okay, so what PCR is going to start with is that you have a region of DNA. So I'm just going to pretend it's like that. So you have DNA that is double-stranded, right? You heat this up, and then you add in these little primers, which are little DNA strands. And what these are going to do is hook on to a region that is specific to these primers. So these primers are made of nucleotides that are going to match this section right here and this section right here. What happens after that is that DNA polymerase is going to come. So DNA polymerase is like a big, it's a big protein enzyme. So imagine it's this big green thing, and it's going to lay a complementary track of DNA like this. See that? And it's going to do the same thing up here. Oh, see, I told you this is going to get hard. And then at this stage, what you have is two completely new. Oh. See, I told you this was going to be ugly. What you have is two new copies of the original section that you used your primers on. You have two regions of this DNA. So remember up here, this region is like continuous. This is in the genome. It goes off in both directions, like pretty much for billions of nucleotides, millions if you're looking at a chromosome. So in this case, this process is all going to repeat like 40 times. But that 40 is exponential. So these 2 will become 4. Those 4 will become 16. Wait a sec. 4 will become 8. Those 8 will become 16, 32. And it gets crazy because, 2 times to the 40th, you start getting pretty big numbers. So you ultimately amplify this region of DNA that you selected with these primers right here. Please do watch the real video. I did try my best. So this is just kind of cool. Kary Mullis is the guy that invented PCR. Basically, he just figured out how to change the temperature, in a sequence and timing that would allow DNA to split, DNA primers to attach, and then DNA to elongate, and then put them back at a new temperature, and they would come back together, and then you repeat that process 40 times. He was crazy. He was like an old school surfer dude. He legit did not. I think he cared about science, but he had other interests to say the least. And he only passed away really recently. But his contribution is so big to molecular genetics. This was the first time we could actually target the DNA that we wanted to look at in detail. Without it, we didn't have the tools to do that at all. Okay, another fun story is going to talk about viruses and bacteria, because I do want to emphasize that this class does have a prokaryotic component, but alongside the prokaryotes and bacteria, I do want to talk about viruses. Given, especially our circumstances, Viruses are essentially living chemistry. What you see in this photo right here are bacteriophages. These are viruses that target bacterial cells. What they are doing is inserting their DNA into this massive bacterial cell. This DNA will go on to make proteins that make more viruses. In the process, they will likely kill this bacterial cell. So, as you look at this, these viruses are, as you can tell, attacking this bacteria cell, and it doesn't look good. 1, 2, 3, they're all getting in their DNA. That DNA is going to basically corrupt the bacterial cell, so this is pretty rough, right? But why I mention arms race is that bacteria don't just stop. Bacteria versus viral evolution is one of the biggest fights of life as we know it. And the evolution of viruses is a whole different thing that's super interesting, and how we kind of contend what when nucleic acids become alive, because I haven't even mentioned obviously the huge conceptual fight of are viruses alive. This is a good video, I'll share this again, of a bacterial cell bursting more viruses that after it was infected they're going to come out and infect more cells. But like this stipulates, there is an ecology, there is an evolution in the microverse. And these two organisms are, well, don't call viruses organisms, or I'll get in trouble. These two organisms are pitted against each other. Okay, so viruses insert DNA and corrupt bacteria. What is an adaptation to defend this poor thing, against something so efficient as a virus? First thing, were restriction enzymes. What these enzymes were massive, big protein enzymes that would come in and chop incoming DNA. See how they split this DNA right here? These enzymes did this so that when DNA would come into the prokaryotic cell, the bacterial cell, it would immediately get the chop, like you see right here. Okay? This was a huge defense and these needed to be very specific, so only specific sequences get cut, because these enzymes would exist in the bacteria at all time. You did not want them cutting the bacterial genome. So these evolved to cut specific sequences that never existed in the bacterial genome. So what we do now with biotechnology is a whole different story. We've taken this bacterial defense system and we utilize it to cut DNA at the places that we want to cut it. We basically take one of nature's coolest adaptations and call it biotechnology and patent it, right? We do that a lot, you'll see. What you can do with these sticky ends, as you can see here what the cut has left, these nucleotides can reanneal, is that we can combine DNA from two different sources. This is DNA splicing, for example. This is the beginning of DNA modification and genetic modification of organisms. We'll see that this process is fairly easy to do and highly controlled, in the scenarios that we have now. But one other defense system exists for bacteria that is a little more popular right now, and that's CRISPR. What CRISPR does is it takes what restriction enzymes did to the next level. So up here, a restriction enzyme can only cut and find CTTAA, right? There are specific enzymes for specific sequences, but that's their limit. They can only cut sequences that look right like this and right like this, right? CRISPR can cut and do damage just about anywhere. That's the key with it. Okay, so let's take a look at this first. What CRISPR is a large protein. See that this big gray area right here? This is a big enzyme essentially, and it has these big nuclease domains. See this one right here, this one right here. What these do is cut DNA at certain regions, okay? And this is where it's special and why it's a combined protein and RNA enzyme. CRISPR takes in this sequence right here, it takes in an RNA nucleotide sequence, a big strand. And this is the guide RNA. What this is going to do is inform the CRISPR where to cut, because the end of the guide RNA right here, do you see how this matches with a set of DNA? Okay? The reason behind this is that bacterial cells needed something that they could interchange. They needed a way to cut all kinds of incoming viral strands. That was the birth of CRISPR. What CRISPR does is basically takes any guide RNA, so any RNA that it's ever encountered before during viral infection, and it can slide it into this region right here and then target it with those nuclease domains right there. Okay? It is an interchangeable defense system that can decide when to kill and what to kill at any time. This is the kind of the birth of CRISPR, and it was a years-long effort. I'll give you a little bit to just think about kind of the road that we took to get here. Zhang's lab, and this is me on a science perspective. Zhang's lab got to the moon, basically, is what I'd call it, but do not discount everything that was discovered previously. The shoulders that got to stand on so that we could get to this moment right here. The editing of the genome of mammalian cells. Our cells are no less susceptible to this process, because what we do is we can insert a guide RNA with a CRISPR, send it in, and make sure this guide RNA matches wherever we want in the human genome to change and nick. See right here, we just want to make a change here, or maybe even delete a region. We can tell the CRISPR to do that by giving it a specific guide RNA. There used to be, this is a sad, funny story, I'll be quick. There used to be a system to change DNA called TALENs. And it was really cool because it was like, oh man, we can edit mammalian cells. It's really hard to use. CRISPR is easy to use by most standards. So TALENs went out the wayside really quick, and that was kind of an unfortunate consequence of CRISPR getting used. Okay, this is something I would like you to know, because this kind of goes into why this works. Like we kind of talked about, CRISPR is the bacterial's memory immune defense. It's memory because, let's take a look here at this timeline. This virus is going to insert DNA. This DNA is going to get chopped up and defeated in this case. Now remember, not every bacterial cell survives, but let's say it does. It's going to take this viral DNA and it's going to put it into its genome. And that's what this red square represents, okay? As you can tell, there are other viral attacks that this bacterial cell has outlasted. It's outlasted all 4 of these viral attacks, okay? Because it has a piece of that viral DNA ready to go. This is the genome of the Cas, of the actual CRISPR gene. Remember the big gray thing? So that's represented in these bubbles. So what you do is when a virus attacks, you make, well, let's do that. What it does is that it transcribes these guide RNAs into RNAs like this, and they form little hairpins like this. We'll get into those structures. It takes them over here and it chops them, and then it also makes actual CRISPR bubbles, or CRISPR proteins like we see right here, and it inserts those guide RNAs into each one, because when the green virus attacks, the green CRISPR guide RNA goes and attacks it, because it's basically saying, I have the green RNA, that green RNA is incoming from bacteria, give it the chop, okay? Same thing with yellow here. It remembers what the yellow virus genome looks like in this case, and it's ready to attack it. I hope I don't draw on this too much, and I'm linked to some really good videos on CRISPR and what it means for us, but even just what it does in bacteria is amazing. The fact that we didn't even recognize that bacteria had a memory immune system. Okay, CRISPR can act in 3 different ways, because only in the last few months did we find a third way. I'll talk about it, but these are the main 2 things you should know. We can direct it to make a break, okay? In a lot of cases, this break is going to occur and cause something called non-homologous end joining. This is going to damage the gene. Sorry, I got that up. This is going to damage the gene because it's not a very good repair mechanism. This red right here represents that the protein is going to be truncated and ruined, basically. Sorry, we got some thunder here. Okay, this is where we start changing things. When we can induce what's called homology-directed repair, we make the nick here, but we also give CRISPR a new template. And our genome is going to repair based on the template, not based on the old green sequence here. Okay? So now we can change or insert or delete specific regions in a gene and change it. And I can't tell you how big of a deal this is. And I know all of you have grown up with, CRISPR is this mainstream thing. This is not a mainstream thing. Changing DNA is not something that has ever been possible. We can manipulate organisms. We can kind of manipulate, straight up genes and how much they turn on or off. This was never possible. We were never able to truly specifically decide how to modify an organism. We've done it with, plants and animals that we've domesticated and bred for specific traits, but we didn't go in and direct that evolution, right? Because now what we can do is induce changes here, which means changes here and changes here. And remember, when you change proteins, you change the phenotype. They are what dictate what happens in the cell for the most part. When we change bacterial genomes, it's even easier because prokaryotes just make copies of themselves. They just propagate even more. So you can have way more like changes as you go with a bacteria because it's going to just make lots of clones of itself. Okay. And this is where we get to essentially where we're headed. The science in this case does not inform us what is right and wrong. We can't find meaning from CRISPR. It's not going to teach us that. We can't find rationality behind CRISPR. It's not going to teach us that either. We know empirically what it can do, but it is up to, you as a scientist, medical professional, anything, to find that meaning in yourself. And it's, you can't turn to primary lit to do that. In a lot of cases, you have to turn to literature. You have to turn to history. When something this large and groundbreaking comes out, there's a lot of questions. Okay, cool. Let's take a look at some more cellular inheritance mechanisms. This is a good time to pause if you want. I'd say that we're well past halfway. I probably spent too much time on CRISPR. It's just super interesting. So, cloning. This was CRISPR 10 years ago, like, oh my God, we actually figured this out. So, in the case of inheritance, we have the technology to essentially take basically a fertilized donor egg from a sheep right here, right? We're going to remove the nucleus, which means we're going to remove and scrub the DNA. And then we're going to fuse this scrubbed DNA cell with just the straight up regular cell of another sheep. Remember, this cell is not a sex cell here, right? This is just a regular cell with a nucleus that contains all the genome. So remember, every single one of your cells contains the entire genome. All 22,000 genes, all 4.7 billion nucleotides. So same genome, basic genome between these two, but now we're taking somebody else's genome and putting it in somebody else's egg, in this case. This egg is going to fuse, embryo occurs, you implant the embryo, this is important, into a sufficient surrogate. This is where the cloning reaches most of its limits, but in the case of sheep, which is a mammal, you can clone something. And as you can tell over here, I often pose this horrifying question to my classes. Barbra Streisand cloned her dog 3 times. Listen, I teach the domestication class. I've lost 2 dogs in a small, tight-knit family, and it's devastating. This is very, very difficult to see because this process right here, this looks nice on a PowerPoint. What I usually ask is, these questions up here, so take some time, you can pause the video right now and kind of think about it, like, hey, you know what, like, would you do it? I've had people say yes, you really you want your dog back, right? You want your pet, anything. And, but don't forget when you're answering this question, these are mammals, humans are not special by any means when it comes to some of these processes, and that's also kind of horrifying. So, if you're thinking about this question, this is the next question I pose. Do you think that these 3 clones were the original 3? Were these the first 3 to work? The answer to that is usually no. Science is a very difficult process fraught with failure. And I can almost guarantee you that by giving them the original DNA to make clones, there could have been many, many, many tries to make a healthy organism from this DNA. And also remember, this DNA was like 9 years old already. So these poor pups came with DNA that's already 9 years old. So they're already going to be premature. And I would not imagine that these three are the only organisms that made it through the cloning process but did not have some kind of horrifying dysfunction, to say the least. That's why ventures that do try to clone animals or pets usually fail out really quickly, because the welfare of the animals is so poor. So again, this draws in that we have a nice clean PowerPoint slide over here. We empirically can do this process. This process does not inform us, should we do this process? On what animals should we do this process? You know, should this be a private industry available for this process? None of the science informs you on those questions. So that's what I want you to think about in this class, because this subject is getting into that territory, okay? Okay. A little bit bleak, not gonna lie. Okay, so let's go into meiosis and go into some final stuff here and kind of think about that. So again, advanced class. When you have a big chromosome, which is millions of base pairs long, you go through S phase and you actually make 2 copies. Remember, these are literal chromatids. These are straight up copies, at least in your, at least in your mitosis, right? And these are going to split both ways, this way and this way, and you'll get 1 chromosome in each of your cells, right? So sometimes this process messes up. So we know that meiosis has 2 stages, one where you should be, so here, this is a nice normal section right here. Usually you are going to split, so these are 2 different chromosomes in this case. In this case, these 2 accidentally both came this way, which means that now with this egg right here in this final meiosis 2 split, you're going to have 2 different copies of this gene, of these genes right here. Same with this one. So what that is going to lead with, or basically end up with, is that when the other cell comes along with 1 chromosome in it, see how this is going to come in here? That's why you get trisomy, is that the normal cell is going to come in, combine with these 2, and now you have 3 of the same chromosome. That causes an imbalance, and that's why you see dysregulatory events, or sometimes, just no embryo forming when you have trisomies or monosomies, okay? It's even worse over here, like you see, these gametes don't have anything in them at all. So this can happen in meiosis 1, where it's a lot more devastating in this case, usually because you end up with more bad possibilities. It can happen in the 2nd stage too, where in this case, these 2 chromatids right here don't separate. So in that case, you have these 2 possibilities. In most cases, though, well, yeah, there isn't much about that. In this case, these events can cause a lot of different things to happen. And one thing that we'll see in this class is that sometimes you don't need whole chromosomes coming with. Sometimes it's just little segments that accidentally come with, right, or get deleted. So we'll see that as we investigate that a little more in more detailed stuff. Oh, shoot. Yeah, I should have just gone to this slide. So non-disjunctions can lead to, okay, now you have a double X, fly in this case over here. So now you have 3 copies of a specific gene. As you can see here, this one has 3 W chromosomes, or sorry, 3 X chromosomes where it shouldn't. And what you find right here is that this egg is not going to give any sex chromosome in this case because it went over to this one accidentally. You see that with the Y chromosome in flies, it needs one of those X chromosomes. This is what we call embryonic lethal. If you're born without a specific region of chromosomes that contain genes that you need for development, you're not going to develop. That's it. You're done. In the other case, you can sometimes see really cool genetic distinctions in these organisms that have this disjunction event. This is actually something, this disjunction, that we actually sometimes try to induce in plants so that we can get more copies of specific chromosomes into a genetically modified plant. Okay, so this is mitosis. I just wanted to briefly touch on this to make sure you feel comfortable with this. We'll focus way more on this, and especially, obviously S phase when we look into DNA replication, and especially as we kind of look more into cancer throughout the class. But for now, I did want to cover mainly, basically, reproduction. This is always a good question to ask yourself, which is, here are these two, like, fairly distinct forms of life, essentially, and how they pass on their genetics. A bacterial cell is going to pass on clone genetics all over the place. That's a pretty huge advantage if you consider that maybe this cell right here has a great advantage in the present environment, right? That's awesome. The key strength, though, of sexual reproduction is that you produce offspring that are unique each time. These are all clones. These are unique. We'll talk about how bacteria can diversify their genome later, but the key thing with sexual reproduction is that you produce very distinct offspring so that as your environment changes, you may have at least one offspring that makes it. And your offspring will be evolving, changing, and bringing new traits to the table, basically, that maybe even sometimes neither parent had or was expressing, but in this combined scenario, the offspring can. Again, this is a very good overview of basically what human reproduction is going to look like at this level. So in this case, one of the father's haploid cells is going to meet one of the mother's haploid cells. You know from independent assortment, from Bio 101, genetics, that the mom has her own 46, for example, and those 46 are made of a pair of 23 and 23 from her father and her mother. But you can't tell which one is which in here, because remember, when they align on the metaphase plate of independent assortment, I'll go back up there, you can't tell which one is going to line up where. So some of what you're passing on is from your mom, and some of what you're passing on is from your dad. And it's random, essentially. That's independent assortment, is that when meiosis starts, it's going to split both of these ways. But the way, if it's really going to be random if you give essentially the homologue from your father or the homologue from your mother. Okay. This is just a fun way to visualize things. And take a look here. And so mathematically, it pretty much works out that you're about 25% of each of your grandparents, or at least blueprint-wise. Now remember, DNA is not the full story, like we've said. But that's about where the math usually works out. Okay. So one thing that this doesn't show is that these regions of chromosomes are interacting. These are crossing over. Remember, some blue is going to go over to red, so like some mother and father genetic material is going to exchange, and that's even going to further diversify what comes on this section and what comes in over to this section. Okay. So this is also another good way to visualize that. Essentially, it's a random combination of if you give away the paternal pair or the maternal pair in either split, right? It would be incredibly rare. So an 8 million chance if all paternal lined up on one side, all 23, and all maternal 23 chromosomes randomly lined up on the other. That happens, as you can tell, because 8 million is 8 million, and this reaction happens in your body a lot, but it's very rare, incredibly rare. Okay. This is kind of neat. This is how we ensure sterile offspring. This is a wild banana. Those are seeds. They are basically like buckshot, and they will break your teeth as far as I've heard. What we made and what bananas really have, these are 3 genome bananas. These cannot independently assort because if you're up here, which side is going to have 2? Is this one going to have 2? Is this one going to have 1? Is this one going to have 2 on this side? Oh, but it's only going to have 1 on this side. So you see this unequal odds even distribution here. That means that these are never going to form proper gametes because they're never going to be equal. You can never get a nice combo of 2 in this case. So another way that we take advantage, like I said, of non-disjunctions is that we can add a lot of genomes to some plants. Some plants can take this. What that happens is that you got more genes, you're going to produce more nutritious proteins that we eat. Now, I've heard that the wild strawberries actually taste still just amazing. They just didn't get basically as beefy as these ones did. Okay, so we've all seen this before, so this is very good to make sure that you understand why it's such a random assortment of genetic material that you get passed on. So even if, all the while you're independently assorting whether or not you're granting, one cell during meiosis, the maternal or paternal, they're also interchanging at all these spots. So this is the most common visual that you usually see is that, you exchange an allele, and that's, these two are going to split, for example, even later. What I can tell you is that essentially these two, and I would draw this on a chalkboard if I could, these two are essentially laid on top of each other, and all homologous regions are interchanging all at once. Tons of interchanges, right? Tons of regions of interchange. Look at that, just like zipper basically. So ultimately, yes, you're aligning maternal and paternal, but there's so much mixing about that the alleles that these, the haploid cells that you're going to get are going to have, there's a huge diversification of that, just massive. And you can't really put that on an illustration, how many times this crossing over actually happens. Okay. This is super interesting. I don't know if I'm going to share this video with you. So you can have hybrid animals. In a lot of cases, so a horse and a donkey can make a mule, right? And this is good. So you have one genome from the mom or dad, one genome from the mom or dad here, and so that makes a working organism. Okay. The problem starts because the crossing over between this mule. Oh wait, no, sorry. The crossing over in this mule. When you're crossing over, see how these have the genes are in the right regions. Like B is in the right region, A is in the right region. In the mule, these alleles are not in the same place. This A might be down like here or something. And so crossing over right here is just blank, and A is down here and there's no good crossing over. You can't go up here. It's all a mess. Crossing over essentially fails in sterile hybrids because the genomes of this hybrid from the mom and the dad they're just too different. So that means you usually never get a haploid that is functional because it doesn't come complete with enough genes to make an organism. It's still really interesting video though. Okay. Fungal division is just super cool. Fungal division can go between asexual reproduction when the environment is kind of harsh, or you just want to keep making clones of yourself until you need to, diversify. And then you can go right back to diversifying through sexual reproduction. And you can combine with more fungi and like diversify and have new offspring. I just think that's awesome. Some protists do this too. It's a very cool it's a very huge survival mechanism. The ability to say, hey, I want to stay the same, or no, I'm going to diversify now. Not, even mammals don't have that option. Okay. So this is the last stuff. This is more prep for the next time. So you can quit here and do this with the next video if you want. This is just going to kind of cover how we start figuring out that DNA is the heritable material, because we did not know that before. You've all seen this experiment for the most part. If you have a virus within, within a protein capsid, it's going to kill the mouse. If you just have regular old DNA that is not encapsulated in this case, the mouse is going to live. This is just a control right here. The mouse is going to live if you kill the strain. And also, the mouse is still going to die if you put in the encapsulated and non-encapsulated strains. But the key here is E. The key is that if you include the DNA that you need to kill, that basically is the viral killer basically, but you include it with the non-encapsulated strains, this can still kill the mouse. And we knew that DNA was the material of this virus after this experiment. Okay. This is a very good image as far as it goes to kind of showcase the central dogma. DNA is the blueprint. DNA is the safest molecule you can store information in. The A's, T's, C's, and G's that are copied into RNA, which goes and ventures out into the danger of the cell, and this will be translated into functional protein. During this period, you also have to keep and replicate your DNA. You do not replicate RNAs, you do not replicate proteins in new cells. You pass along a copy of your DNA. It is the more important molecule when it comes to inheritance. Reverse transcription is something that where a virus actually carries its genetic material as RNA and it goes back to DNA to integrate into the host. That's a special case. This is just a good, basically a great look at how RNA polymerase actually looks as a massive protein. It basically opens up DNA right here and copies this section right here into this green RNA in this 5' to 3' direction. So it's really, really good. Next, this is translation. This is going to take our RNA. Oh, sorry. This is going to take our RNA and it's going to read that triplet code, 3 by 3 by 3. And each of these 3 is going to count as an amino acid. And this huge amino acid is going to go on and form massive proteins, like RNA polymerase. Remember, RNA polymerase is a protein, all the enzymes, all the defense systems, all the protein transporters of your cell, they're made of polypeptides. And that's why this is called translation, is you're going from the very non-functional language of nucleic acids. Remember, these nucleic acids don't have a ton of function. They're not very dynamic chemicals. They can't grab stuff, hold stuff, conformational change as much. We'll see when they can, but it's very minimal compared to polypeptides. You translate into this language of proteins. I've never gotten a triangle before. Interesting. And in that case, this is where the functions of the cell really lie. So this is the end. This is what we'll cover next time. Basically getting this blueprint and causing it to act.